Good morning, my dear students. Today's lecture topic is on drug development and how we are going to evaluate the new drugs. So, what do you understand by the term development and evaluation? Development is we will be developing or newly creating a drug. Then, after you just created a drug, that doesn't mean it is safe in use in general population. So you have to evaluate whether this drug will provide or create any lethal or bad toxic effect. Because while you are developing the new drug, you will check how it works, what is the dose you have to give, what is the T half and everything you will check. But very important in evaluation is there any toxic effect? If the benefit overweigh the risk, if there's a risk, if the risk is fatal, like that, we will check it. So to be success, a new drug must make it through 5 steps. You are creating, you want to create a drug. And what is the purpose of creating drugs by most of the pharmaceutical companies? They want to make profit. The pharmaceutical company don't love general population. They want to make profit. So with the competition with the other companies, they make better drugs. So if you are going to make a better drug or drug, and then you want to release that to general population, there are 5 basic steps. One is discovery and development. For example, if there is a problem, for example, you are going to find a drug for the diabetes. Then you know the physiology behind how insulin works, how glucagon work, and everything. Then you decide, okay, the problem you found the problem is with the insulin receptor insensitivity. So you are going to stimulate the receptor activity. So you are creating insulin receptor sensitizer. So that is how you discover what you are going to create. Then with the very detailed, complex studies and investigation in your laboratory level, you will create a compound, a lead compound. Okay, that is the development. Then you just read about or go through, work about the basic idea of the lead you created. What is the mechanism of action? How it works? What is the dose I may need to give? What is the interval? Blah blah. You have to work on that. Then you come for the preclinical research. Now you have an idea. Then with this idea and some other formalities you have to do, I will explain later. You will come to the preclinical research. That means you do research on animals. Then if it is success, and if you believe the results are favorable to test that particular drug on human with the approval from certain authorities, you will do clinical research. Then you will get the FDA review. FDA is Food and Drug Authority. Then you will look for a safety monitoring of the particular. So these are the basic things I have explained. Screening and discovery, then preclinical development, then you do studies in target species, then you need to get registration, then you do manufacturing and marketing, then application in human and veterinary medicine in the large scale. The 5 drug development phases, what we have again gone through, just go through this. Step 1, discovery and development. Research for a new drug begins in laboratory. Typically, researchers do many tests and found a molecular component or compound that is suitable to become a drug or that will make possible beneficial effects against a large number of diseases. That's how the discovery happens. At this stage, thousands of compounds become potential candidates. But after early testing, however, a small number is progressed to the further study. That is usual things happening, right? Even in the life. Then the development. Now, once the researchers identified the promising compound, then they conduct a lot of experiments to gather information. How it is absorbed, distributed, what are the potential benefits, what is the best dosage, what is the best way to give, either mouth or injection, what are the preferable or possible side effects, blah blah. You have to get an overall good in detail information. So this is what they say. There's a target selection, there's a lead discovery, then medicinal chemistry, you work it out, and in vitro studies. This all happen in the laboratory. Preclinical research. As I told you, the purpose of preclinical research is largely to assess whether a compound has the potential to cause serious effect or serious harm. Whether that particular lead compound or leading compound is causing serious effect or harm on humans or not. So before testing in a drug in people, researchers they will check their toxicity. There are 2 types of preclinical research. One, either you will do in vitro or in vivo. To do that, there should be a good laboratory practices and there is a certain guideline and you have to follow that. So these are the basic requirements for conducting preclinical studies. You have to have a study conduct. There should be a personnel, facilities, you should have equipment, there's a written protocols, operating procedures, study report, and lot of things. Usually, the preclinical studies are not very large, but however, the studies must provide detailed information on dosing and toxic levels. After preclinical testing, Researchers review their findings and decide whether the drug should be tested on human or not. Then clinical research. Once a drug is tested on animals, the preclinical research, and once we found the dosage and toxicity, if it is okay to use on human, we will use it. But remember, the preclinical research, that means what we do in the rats or animals, answers basic questions about drug safety. But these are not a substitute for studies to, that means these are not substitute for studies of ways that drug will interact with the human body. Whatever the interaction drug is done in animal body, you can't directly apply that to human. Before you give that to human, you have to do research on human, and you have to again go through the safety profile of that particular drug on human. Okay, so this process we will call investigational new drug process. A process they must go through before clinical research begins. As the developers design the clinical study, they will consider what they want to accomplish for each of the different clinical research phases and begin the investigational new drug process. So this process is contained the clinical study phase, designing clinical trials. You have to design it. Then you do the clinical research phase studies. That means you have to decide how you are going to do. Then the investigational new drug process, asking for FDA assistant, FDA and IND review team approval. Designing clinical trials. Researchers design clinical trials to answer specific questions related to the medical product. These trials follow a specific study plan called a protocol that is developed by the researcher or manufacturer. Before the clinical trial begins, researcher review prior information about the drug to develop research questions and objectives. Then they decide who qualifies to participate, how many people will be participated, how long the study should last, whether there will be a control group, or is there any other option to limit research bias, how the drug will be given to patient and at what dosage, and like that they will be go through. The clinical trials follow a typical series from early small scale phase 1 studies to late large scale phase 3 studies. This is very important. Clinical research phase studies. There are mainly 4 phases. Listen very carefully. The preclinical research. What we do is preclinical trials. In the we check for the dosage and safety in animals. Once we check it, we will get approval from testing in humans. After the preclinical trials, what you do is you get all the data, you analyze your results, You think, okay, the dosage is good and the toxicity are very low or minimal. Then you get a permission from authorities to test that drug on human. That is very important. Once you get the approval to test that particular drug on human, what you do is in the phase one, you will you will select voluntary healthy humans, 20 to 50 or 80 participants. Then you perform or the research. Mainly this point you will select healthy individual. But in few rare cases, few occasions, even the disease individual we use for phase one clinical trial. Then phase two, you perform that on more participants. Then phase three, if these become successful, you do phase three in 1000 to 3000 participants. They are disease individuals. The phase three can happen in the ward as well. Then the results you will submit for FDA approval. Then FDA will review it and they will confirm the safety and effectiveness of the drug is good. Then only drug is approved. Once the drug is approved, it doesn't mean it is 100% safety to use on human, because these three phases are not enough to study the longer term side effect and toxicity of the drug. So after it approved and released into general population, you have to monitor via drug monitoring, drug interaction or toxic or side effect notification to study or learn about the full side effect or profile of the drug. So it is a very long term process. So the phase one, I explained to you very clearly. You just go through, take few minutes and try to understand this. Okay, I have highlighted very important points. In the after the end of the phase one, approximately 70% of drugs move to the next phase. After the end of the phase two, approximately 33% of the drug moves to the next phase. After the end of phase three, this is after the end of phase two. After the end of phase three, approximately 25 to 30% of the drugs move to the next phase. The phase four is several thousands of volunteers who have disease and condition, and mainly you look for safety and efficacy. Then we will see what is investigational new drug processes. Drug developers or sponsors must submit an investigational new drug application to the FDA before beginning, listen very carefully, before beginning the clinical research. So the investigational new drug is IND. So the IND application, the developers must include animal study data and toxicity. What happened during the animal study, that is preclinical studies. Manufacturing information, how the chemical compound is manufactured and synthesized, or everything. What is the clinical protocol for the studies to be conducted? If there is any data from any prior human research, that means some manufacturer has started the research and in the midway he stopped it due to financial reasons or any other reason. And very important is information about the investigation. Then if you need any assistance, the FDA will assist you before the IND process or during the preclinical, the clinical phases, or once if you want to change design from phase 2 to phase 3 or whatever, at any time of your research, FDA will help you. You can ask from them. Then in the FDA review team, to analyze the IND application, there are set of different members with different qualifications. Project manager, medical officer, statistician, pharmacologist, pharmacokineticist, a chemist, and a microbiologist. Once the approval is given by the FDA, they usually review within 30 days and will give a approval. Either they will approve to start the clinical trials, or they will ask to hold or to stop investigation due to few other reasons. One reason is investigators are not qualified. So proper qualified investigators are needed for this drug development. You just go through, these are just theories. Okay, you can have time and you can read. Then now so far we have seen the drug development process. There is 5 stages. One is discovery and development of the lead compound, the leading compound. Second thing is preclinical studies, but we are performing on the especially rat in the laboratory condition. Third thing is after the laboratory preclinical studies, you will send the IND application. Once it is approved, you do clinical studies. Once the clinical study is finished, again with the data, you look for FDA drug review. If a drug developer has evidence from its early test and preclinical and clinical research that a drug is safe and effective for its intended use, the company can apply to market the drug. The FDA review team thoroughly examines all the submitted data on the drug and decide whether or not to approve it. So these are a new drug application, how it should, how the application, what are the content the application should contain, how it's going to apply, all the details are there, just read it. Once the FDA determines the drug is safe and effective, then they want to refine the prescribing information. This is referred as labeling. Okay. So they want to refine the prescribing information. Labeling accurately and objectively describe the basis for approval and how best to use the drug. And step 5, the post-market drug safety monitoring. This is very important. Even though I told this earlier also, even though clinical trials provide important information on a drug safety and efficacy, it is impossible to have complete information about the safety of a drug at the time of approval. Despite the rigorous steps in the process of drug development, limitations still exist. Therefore, the true picture of a product safety evolves over the months and even years that make up product lifetime in the marketplace. FDA reviews reports of problems with prescription and over-the-counter drugs and can decide to add caution to the dosage or usage information, as well as other measures for more serious issues. So with that, we finish how to develop a drug. And we'll go through few ethics in clinical studies. In history, there are Nuremberg Code, Declaration of Helsinki, Belmont Report, and few other things. What is Nuremberg Code? We'll go back to Second World War. At the end of the Second World War, the Soviet allies won the war against Nazis. So they want to bring the Nazi in front of justice. They want to punish them. So there were series of 13 trials carried out in the Nuremberg. This is one of the major city in the Germany. Okay. In that Nuremberg trials, they found out there are a lot of medical and non-medical research happened without proper ethical clearance. They were against patients or subjects wish. Patients or subjects are used without their permission. Even though they don't want to get into the research, they forcefully get into that. And there were a lot of harmful gases and substances check on the human. The history says. So they have created a Nuremberg code that to prevent the patient or subject harmfulness or to maintain or safeguard their ethical rights. So Nuremberg code contains 10 things. This is not the lecture for that. So just go through that. No, there is something called Nuremberg Code and the Declaration of Helsinki. This is a wonderful story. There's a drug called thalidomide. Thalidomide is a drug initially found to use as a tranquilizer and later they used to relieve anxiety, fear and sometimes the morning sickness. Those drug is never tested on pregnant women, but in pregnancy women get morning sickness, so they prescribe it on pregnant women and it worked well on them. The morning sickness settled, but the babies born for those mothers were without the limb and the hand and leg. That means there is only budding part of hand if they are not the proper hand. So that is the serious effect. Then around 10,000 children were seriously disabled and 2,000 were died. So these are the large numbers. So the authorities decide to create very strictly followed codes or proper way to do a clinical trial. So that is called Helsinki preparation or declaration. Go through this. So whenever there is harmful things happen to the subjects or offspring, or whenever the safety is not there, the thinking of ethics is come to the mind. That is how ethical codes or ethics in research is developed. We always learn from mistakes. That's how it also develops. So ethics of research in humans, there are 5 things, basic things. Autonomy, beneficence, non-maleficence, justice, and informed consent. Autonomy is the people have the right to choose for themselves whether they want to participate in a research or not. They should have the right. You can't force them. Beneficence. The doctors have a moral obligation to ensure they do the best for the patient, maximizing the benefit and minimizing risk. Non-maleficence. Doctors should never allow a patient to put at risk. That means doctors know this drug can cause harm on this patient, and they know it. And okay, we'll see and do. You can't do that. Okay, that is non-maleficent. If you strongly believe it's going to put this fellow at a risk, don't do that. Then informed consent, you have to explain the volunteers. These are the things we are going to do. Okay, and you have to get them know what we are going to do, what may be the possible risk of that, what is the usage for the society, and there are a lot of things in the informed consent. It should be taken by the investigator, the language of the participant. Okay. In the case of young children or mentally retarded patients, consent should be obtained from their guardians or legal representative. Imagine if the subject is illiterate, the person doesn't know how to read or how to understand some language. Then a legally accepted representative or an impartial witness needs to be present when you are obtaining informed consent. So there are techniques to avoid bias, randomization, and blinding. What is bias? In research, bias occurs when a systemic error introduced into sampling or testing by selecting or encouraging one outcome or answer over others. That is systematic error. Bias can occur at any phase of research, including study design or data collection, as well as the process of data analysis and publication. So to avoid that randomization and blinding, yeah. Sorry, I'm having a very cold and sneezing. Randomization, random allocation of patient to control an experimental group so that allocation bias is minimized. That means you randomly allocate patient for the control group and for the testing group. There are various type of randomization and blinding. You control a control device to prevent bias influencing research. There are designs used in clinical trials, open trials, single blind, double blind. What is placebo? It's a dummy medicine. So what are the uses of that you can go through. And the pharmacovigilance is the process of identifying and responding to issues of drug safety through the detection in the population of a drug effect, usually adverse things. So techniques of pharmacovigilance are voluntary reporting, prescription monitoring, medical records, and population statistics. So there are few strengths of evidence for a clinical research. The top one is systematic reviews and meta-analysis. Then randomized clinical trials with definitive results and non-definitive, cohort, case control, cross-sectional studies, case reports, etc. This is the diagram or hierarchy of the evidence of the research. There are few reference. I hope, yes, you all are our brilliant students. You have understood very clearly. If you have any doubt, you can contact me via WhatsApp or email. As I always say, may peace with you all. Thank you very much. Have a nice day, my friend. Yeah.